There's a particular kind of stillness that comes with being unwell, the way the world continues at its usual pace and you just watch it from somewhere slightly behind your own eyes. Everything feels a little distant tonight. The throat is doing that dry, tight thing it does, and breathing takes a bit more attention than usual. Welcome to the Sleepy Biologist. I'm glad you're here, even if here is under a blanket with a box of tissues nearby. This channel exists because of the people who keep coming back, the members and supporters who quietly make this space possible. And I think about that especially on the nights I almost didn't record. There's something oddly fitting about talking tonight, congested, a little slow, about the way a forest exhales, because that's what's happening right now, somewhere above the tree line, not dramatically, not even noticeably if you were standing beneath it. The topmost leaves of a forest are releasing gases into air that will carry them farther than the tree's roots will ever reach, and the tree has no knowledge of this. None of the apparatus involved, not the leaf, not the pore, not the gas molecule drifting upward, contains anything like awareness. It is chemistry, patient, continuous, responsive chemistry. The pores responsible are called stomata. They open in response to light, to the concentration of carbon dioxide inside the leaf, to water pressure within the cells that guard them. 2 kidney-shaped cells flank each opening, and when they fill with water and swell, the pore between them widens. When they lose water, it closes. It's strange, when you slow down enough to think about it, that something as invisible as a gas concentration can trigger a mechanical response in a living cell. No signal was sent anywhere, no decision was made. The cell simply changed shape because its internal chemistry shifted. You've probably felt something a little like this today without naming it. The way your own airways have been making decisions without you, tightening, swelling, producing things you'd rather they didn't. Your body responding to an irritant it detected before you consciously registered anything was wrong. There's a version of that happening in every leaf at the canopy's edge right now. What exits through those stomata is mostly water vapor, and then carbon dioxide. The carbon dioxide the leaf has been producing as a byproduct of its own cellular respiration. But in daylight, the leaf is also running photosynthesis, pulling carbon dioxide in through those same pores and fixing it into sugar. The net direction of carbon dioxide, whether more leaves the leaf than enters or more enters than leaves, shifts depending on light intensity. In the early morning, as light increases, photosynthesis accelerates past the rate of respiration. The leaf becomes a net absorber. Carbon dioxide is being taken in faster than it's being released. But this doesn't happen instantly. There's a lag. The chemistry has to catch up with the light. Enzymes need to activate. Chloroplasts, those flattened disc-shaped structures distributed through the leaf's interior cells, begin orienting toward the incoming light, actually moving within the cell, repositioning to intercept more photons. That repositioning takes minutes, not seconds. And all of this is happening in the canopy, 30 or 40 meters above ground, in leaves that are constantly adjusting to a light environment that changes as clouds pass, as neighboring branches sway, as the sun climbs incrementally higher. The carbon compounds that do escape, the water vapor, the residual carbon dioxide exhaled during respiration, rise. Warm air above the canopy carries them upward. Some will travel kilometers before coming down. Others will react with other atmospheric compounds and return to the forest in rainfall weeks later, absorbed through roots into a system that has no memory of the original release. Something about that is hard to fully hold on to. Carbon that left through a single leaf pore on a Tuesday morning in one season, returning dissolved in rainwater the following spring, entering through root hairs in the dark of the soil, moving upward through xylem into a new leaf that won't open till April. The same atom, a completely different journey. The canopy doesn't track any of this. It resonds, releases, absorbs, adjusts, and then the light shifts again and the whole calibration begins once more. Every square centimetre of leaf surface doing its own version of this simultaneously across the whole crown of the tree, across every tree in the stand, across the full extent of the forest. There's no coordinating signal. No message passes from one leaf to another saying the light has changed or that the air is drier today. Each leaf responds to its own local conditions, and yet, when you look at a forest canopy from above, if you've ever seen one from a plane window or from a high ridge looking down, it moves together. Not in unison exactly, but with a kind of coherence. A slow collective breathing that emerges from millions of entirely independent responses. I find it genuinely hard to explain why that's so calming. Maybe because it's a system producing order without needing to enforce it. The leaves at the very top are the first to register the morning. By the time light reaches the understory, the canopy has already been running its carbon chemistry for hours. The forest floor lives in a different time than the canopy above it. Not separate. The carbon moving downward through the phloem connects them, but delayed, filtered, quieter. And below all of it, below the bark and the cambium, and the root tips reaching into the dark, there is soil that has been accumulating carbon for longer than any of the trees above it have been alive. Carbon that arrived through different leaves, different roots, different organisms, carbon that has been waiting in some form for centuries. The canopy releases without knowing, but what it releases doesn't disappear. It just moves somewhere the canopy can't see. There is something under the forest that the forest itself doesn't know about. Not hidden exactly, just older. Older than the oldest trunk, older than the root system that has spent decades threading through it. Older than the particular arrangement of species that currently constitutes this forest. The soil beneath a mature woodland is a kind of record that nobody asked to be kept. And most of what it contains arrived here through processes that no longer operate in quite the same way. That's the part worth sitting with for a moment. When a leaf falls and decomposes, it releases its carbon relatively quickly. Within a season or two, much of it has been processed by fungi and bacteria and returned to the atmosphere or incorporated into microbial bodies. That's the fast carbon. The part of the cycle that moves at something close to human timescales. But not all carbon moves that way. Some of it moves downward. Dissolved organic compounds leach through the upper soil horizons with rainfall, passing through layers of increasing density and decreasing biological activity. As they descend, they encounter mineral surfaces, clays, iron oxides, compounds that carry electrical charges along their surfaces, and some of the dissolved carbon binds to these minerals, not loosely, in ways that are genuinely difficult to break. This is carbon that has, in some functional sense, gone still. The layers where this stabilized carbon accumulates are called mineral-associated organic matter horizons. The name isn't important. What's interesting is what it means. That there are places in the soil beneath your feet. Beneath the forest, beneath the root zone, below where most organisms bother to go, where carbon is being held in chemical bonds that have outlasted multiple generations of the trees above them. Some of this carbon was fixed by leaves that fell before anyone living has any memory. Before the current forest composition established itself, before the climate settled into its present pattern. There are soils in old-growth forests where the deep carbon has been dated to several thousand years before present, sitting unchanged while the surface above it cycled through drought, regeneration, and succession. It didn't go anywhere, it just stayed. The microbes that live in these deeper layers are not like the dense, active communities near the surface. They are sparse, slow metabolizing. Some of them divide so infrequently that their generation time is measured in years rather than hours. They are working, technically working, but at a pace so subdued it barely registers against the furious biochemical activity happening in the top few centimeters of soil above them. What they encounter down here is largely resistant to them anyway. Lignin, the structural compound that gives wood its rigidity, is one of the more stubborn molecules in the carbon cycle. Most bacteria can't touch it. Only a narrow group of fungi, white rot fungi primarily, produce the enzyme systems capable of breaking lignin's irregular polymer structure apart. And even those fungi work slowly over years, not days. When lignin-rich material is buried before that decomposition is complete, its carbon can persist far longer than the organisms that built it ever lived. There's something I keep coming back to, which is that the soil is full of biological effort that simply didn't finish. Processes that were interrupted by burial, by cold, by the absence of the right organism at the right moment, and then quietly preserved. Not preserved intentionally, just not undone. Below the rooting zone, below even the deepest taproots of the largest trees, the soil is also receiving carbon from above in the form of root exudates, compounds that roots actively secrete into the surrounding soil, which then leach downward with water movement. The tree doesn't intend to feed the deep soil. It's managing its own rhizosphere chemistry, adjusting the pH around its roots, releasing compounds that help solubilize minerals it needs. The carbon that escapes downward is, from the tree's perspective, a byproduct, but byproducts accumulate over centuries. There is also carbon arriving from a direction you might not expect. Mycorrhizal fungal networks extend deep into mineral soil layers, following fractures in rock and pockets of moisture. When these fungi die, their cell walls, rich in a compound called chitin, decompose slowly, contributing carbon and nitrogen to layers that receive little direct input from plant material. Chitin is resistant, not as resistant as lignin, but slower to break down than most cellular material. The bodies of dead fungi are a significant and often overlooked input into the deep carbon record. It's one of those things that doesn't make it into simplified descriptions of the carbon cycle, which tend to focus on the dramatic exchanges: photosynthesis, respiration, decomposition. The slow accumulation of fungal cell walls in deep mineral soil isn't dramatic, it's just steady. And steadiness over geological time builds up into something considerable. The forest above has no access to most of this stored carbon. The roots can't reach it. The chemistry that locked it into mineral surfaces isn't reversed by anything the living forest produces at its normal operating pace. In that sense, the deep soil and the living canopy are running on different schedules entirely, connected in origin, but no longer in active exchange, which means the forest is, in some respects, built on top of a record of itself that it cannot read. The surface is busy, actively cycling. Carbon in, carbon out, organisms consuming and releasing and dying and decomposing across timescales that feel almost urgent compared to what lies beneath. And beneath that, the slower layer, the mineral-bound fraction, carbon that entered a different kind of time when it crossed a certain depth. It's not entirely clear what disturbs this stored carbon most. Temperature increases may accelerate the microbial communities even in deep layers, gradually unlocking bonds that have held for centuries. Physical disturbance, deep tillage, compaction, erosion can expose mineral-bound carbon to conditions it hasn't encountered since it was first buried. Whether the deep soil's carbon is truly stable or only stable within the narrow range of conditions the current climate has maintained, that question doesn't have a clean answer yet, What's certain is that it's there, in quantities that dwarf what's currently cycling in the living biomass above it. The trees are visible, knowable. You can walk among them, measure their trunks, watch them leaf out in spring. The carbon in the soil beneath them is a different kind of presence entirely. Deeper still, past the biological horizon, past the reach of roots and fungal threads, the rock itself contains carbon, ancient carbon, fixed by organisms that no longer exist, in forms that have been compressed and chemically altered beyond recognition. That carbon is not part of the forest's cycle, not yet. The forest sits on top of all of this and breathes at its own pace, largely unaware of what's underneath. Somewhere below the roots, a carbon atom is held against a clay surface in the dark, where it has been for longer than this particular forest has had a name. You already know what it feels like when something in your body wants something it isn't getting. The low-grade pull of hunger before you've consciously registered it. The way your hand moves toward water before thirst has fully formed as a thought. There's a version of this happening underground right now, not in any creature with nerves or awareness, but in a network of fungal threads so thin they pass between soil particles without displacing them, running through the dark in every direction toward things they need. Mycorrhizal fungi don't have wants, obviously, but their behavior, if you watch it long enough, starts to look stubborn in a way that's hard to describe without borrowing that word. The relationship between these fungi and the trees above them is old, genuinely old. Fossil evidence places mycorrhizal associations at around 450 million years before the present, which means this particular arrangement predates forests themselves. The trees that exist now inherited a partnership that was already ancient when the first land vertebrates were still figuring out legs. And it is a partnership in the functional sense. Carbon moves down, minerals move up, but it's not equal, and it's not fixed. The tree produces sugars through photosynthesis, glucose, sucrose, compounds built from atmospheric carbon dioxide and light. Some of these sugars travel downward through the phloem, into the roots, and from there into the fungal threads attached to the root surface or woven between root cells. The fungus receives carbon it cannot produce itself. It has no chlorophyll. It cannot photosynthesize. Without plant carbon, it starves. In return, the fungal network extends far beyond what the root system can reach, accessing pockets of soil where phosphorus, nitrogen, and other minerals are concentrated. Phosphorus in particular moves poorly through soil on its own. It doesn't travel in water the way nitrate does. The fungal threads reach it and transport it back toward the root. Simple enough on the surface. Except that the allocation of carbon, how much the tree actually sends down, isn't constant. It shifts. When phosphorus is abundant in the soil around a tree's roots, the tree reduces its carbon transfer to the fungal partners. The fungi are receiving less payment for a service the tree currently needs less. Whether this constitutes something like negotiation is a question biologists argue about carefully because the word implies intention that isn't there, but the outcome is functionally similar to what negotiation produces. Resource flow adjusts to reflect current need. The fungi, for their part, respond to reduced carbon by allocating less phosphorus transport toward that particular route. Both parties pull back simultaneously, and the exchange quietly recalibrates. What makes this stranger is that a single fungal individual, if individual even applies to something without clear boundaries, may be connected to multiple trees of the same or different species simultaneously. Carbon from a large, well-lit canopy tree flows into shared fungal threads that also connect to a smaller, shaded seedling receiving less light and producing less sugar. Whether carbon genuinely moves from the larger tree to the smaller one through this shared network, or whether the seedling simply benefits from the fungal network's own carbon reserves is still being worked out. The forest doesn't offer clean experimental conditions. I tend to think the ambiguity is part of what makes this worth paying attention to. Not everything about this network has resolved into a tidy explanation yet, and there's something genuinely interesting about a system that's been running for 450 million years and still hasn't been fully read. The threads themselves, hyphae, are between 2 and 10 micrometers in diameter. For context, a human hair is roughly 60 micrometers wide. These threads move through soil, not by pushing through it, but by growing at their tips, extending forward while the older portions remain fixed. The growing tip secretes enzymes ahead of itself, loosening organic material, accessing pockets of mineral-rich moisture. A single gram of healthy forest soil may contain hundreds of meters of these threads. That number is hard to picture. It's easier to just accept that the soil is far more populated than its surface suggests, and that most of what's happening in it is happening at a scale you'd need a microscope to confirm. Speaking of things happening at scales difficult to perceive, I've spent most of today being acutely aware of my own mucous membranes, which are doing their own version of a resource allocation crisis. I'm starting to think I might need 2 blankets tonight. The fungi are fine. I am less fine, back underground. The carbon the tree releases into the fungal network doesn't all go toward mineral exchange. Some of it feeds fungal growth, the production of new hyphae, the maintenance of existing ones. Some goes toward reproduction. Spore production requires significant carbon investment, and some ends up in the soil as fungal byproducts. Compounds secreted during hyphal growth, dead cell walls left behind as the network remodels itself. This leaked carbon becomes food for bacteria, which become food for nematodes, which release nutrients that roots can absorb. The transaction that began as a 2-party exchange between tree and fungus branches outward through the soil community in ways that weren't part of any original arrangement. Carbon allocated downward to pay for phosphorus ends up sustaining organisms the tree will never directly interact with. The part that sits with me most is how the network responds to stress. During drought, when the tree reduces photosynthesis to conserve water, stomata closing, sugar production slowing, the carbon flow to fungal partners drops. Some fungal species tolerate this reduction and maintain their connections, others don't, and the hyphal threads attached to those roots die back. When rain returns and the tree resumes full photosynthesis, it reconnects with whatever fungal partners remained. The network that reconstitutes after drought is not identical to the one before it. Some species are gone. Others that were minor partners before may now dominate simply because they were the ones that stayed. Stress selects, not intentionally, just through the blunt arithmetic of who remained connected when the carbon stopped. Tonight the trees are doing whatever trees do after dark, no photosynthesis, stomata mostly closed, carbon running through the phloem at a slower rate than it did this afternoon. The fungal threads are still active, still growing at their tips, still moving phosphorus toward root surfaces that may not take it up until morning. The exchange doesn't stop, it just slows to match the tree's nighttime pace, and in the soil, in the dark, threads a few micrometers wide are extending toward concentrations of minerals they detected through chemistry alone, in a direction that has no name because nothing down there has ever needed to name anything. The carbon follows the need. It always has. Hold your hand up in dim light sometime, not now but sometime, and look at it long enough that it stops feeling like yours. The skin, the small geography of knuckle and vein, is made of cells that were produced recently. The hand you see is not the hand you had a year ago, not entirely. Cells have turned over, proteins have been replaced, and yet the hand is continuous, recognizable, yours in every sense that matters. The material changed. The form persisted. A leaf works on a version of this same logic, except the timescales are compressed into a single season, and the carbon inside it has a history the leaf itself carries without knowing. When a tree breaks dormancy in early spring and begins producing new leaves, it doesn't start from nothing. The carbon that builds those first leaves, and the sugars and amino acids that become cell walls, chloroplast membranes, the waxy cuticle on the leaf surface, comes from reserves the tree stored the previous autumn, starch packed into ray cells in the sapwood, soluble sugars held in twigs and small branches, nitrogen compounds withdrawn from last year's leaves before they fell and tucked into bark tissue for winter. The new leaf, still pale and soft in early April, is built substantially from material that was already inside the tree when the first frost came months ago. There's something quietly strange about that. You're looking at what appears to be new growth, and in one sense it is. These cells have never existed before, but the atoms inside them have been here, in this tree, through the cold and the dark and the stillness of winter, waiting in stored form for the chemistry to shift back toward growth. Carbon moves through a tree in ways that don't follow a simple path. The phloem, the tissue that transports sugars from leaves to the rest of the tree, doesn't run in one direction. It runs toward wherever demand is highest. In spring, that means carbon flows toward expanding buds, new root tips, the meristems, where cell division is happening fastest. In summer, some of it is pulled toward fruit if the tree is producing any. In autumn, it flows inward, toward storage. A carbon atom fixed by a leaf in June might travel downward into a root, be incorporated briefly into root cell walls, then released back into the phloem when those cells are broken down and remobilized later in the season. It might spend a winter in the woody tissue of a branch as part of a starch granule, then become part of a sugar loaded into phloem the following March, then end up in a leaf that opens in April. The leaf you see in spring may contain carbon that has made 2 or 3 internal journeys within the tree before arriving in its current form. This is the part that the extended metaphor of a library, which you'll sometimes see used for forests and their stored information, fails to capture, and why it's worth letting that metaphor go here. A library stores things in fixed locations. The carbon inside a tree is never truly fixed. It circulates. It gets incorporated, disassembled, reassembled, rerouted. The tree is less a storage system than a continuous negotiation between where carbon currently is and where it's currently needed most. The leaf is a temporary arrangement, an expression of what was available and what was required at this particular moment in the tree's annual cycle. What the leaf does once it opens is begin accumulating carbon of its own, fixing atmospheric carbon dioxide through photosynthesis, building new compounds, contributing to the pool that will eventually feed the whole tree. By midsummer, a healthy leaf is running a surplus. It's producing more sugar than it needs for its own maintenance, and the excess moves outward through the phloem to wherever demand pulls it. But the leaf's own history, the carbon it was built from in April, remains inside it, distributed through its structural components, the cellulose in the cell walls, the proteins in the chloroplasts. These don't turn over quickly. For most of the leaf's life, that foundational carbon stays put, even as new carbon accumulates around it through the season's photosynthesis. So a single leaf contains simultaneously carbon that was fixed this week and carbon that spent last winter in the heartwood. In autumn, before the leaf is released, the tree begins withdrawing what it can. Chlorophyll breaks down, the green fades, and the yellows and oranges that were always present but masked become visible. Nitrogen and phosphorus are actively pumped back out of the leaf tissue and moved into the woody parts of the tree for winter storage. Some carbon compounds are withdrawn as well, though incompletely. What falls is already partially emptied. The tree has taken back what was worth keeping. The carbon that leaves with the fallen leaf is carbon the tree judged, through no judgment at all, purely through the economics of transport costs versus nutrient value, not worth retrieving. It lands on the soil surface and begins a different kind of journey. The leaf that fell last November is somewhere in the duff layer now, softening, its cell walls slowly yielding to fungal enzymes. The carbon inside it is becoming available again, entering the soil food web, moving toward a form that roots might absorb next spring. Some of that carbon will be taken up by the same tree that dropped it, returned through root hairs loaded into xylem water, carried upward, incorporated into new tissue. The same atom, a different leaf, the same tree older by a year, running the same accumulation and withdrawal it has been running since it was a seedling. Your hand in the dim light, cells turned over but the form persisting. The leaf in April, built from winter reserves, fixing new carbon through the summer, giving back what it can before it falls. Both of them continuous, both of them made of material with a longer history than their current form suggests, and both of them right now resting. I remember the exact moment my foot caught it. Mount Airy Forest, Cincinnati, a trail I'd been walking casually, not paying particular attention to the ground. And then a root. Not a dramatic one, not the thick, arching kind you'd notice from a distance. Just a low, half-buried thing that had grown across the path at exactly the wrong angle, and my foot found it before my eyes did. I went down, hands forward, one knee into the soil, face suddenly very close to the forest floor, and I remember staying there for a moment longer than necessary, not because anything was hurt, but because from that angle, face close to the ground, hands pressed into the duff, the forest looked completely different. The root that caught me disappeared back into the soil a few inches away. Where it went from there, I had no idea. That's the thing about roots. You only ever see the part that broke the surface. The rest of it is the decision the tree made in the dark. Roots don't wander. That's the first thing worth understanding. They look like wandering, the way they curve around rocks, cross over each other, emerge unexpectedly on the surface of a trail, but each extension is a response to something specific, a gradient, a concentration, a chemical signal the root tip detected and followed. The tip of a growing root contains a region called the meristem, where cells are dividing continuously, pushing the root forward through the soil. Just behind the meristem is a zone where those new cells elongate, stretching out, which is the actual mechanism of root extension. The tip itself is covered by a root cap, a loosely arranged cluster of cells that gets worn away by friction with soil particles and is continuously replaced. The root cap does something interesting. It detects gravity. Specialized cells within it contain starch-filled organelles called amyloplasts, which settle toward the lowest point of the cell in response to gravitational pull. This settling triggers a signaling cascade that directs root growth downward, or more precisely that corrects growth back toward downward when the root has deviated. It's a continuous error correction system, running without a brain to run it. When a root tip encounters a zone of higher moisture, cells on the drier side elongate faster than cells on the wetter side. This differential growth, more extension on one side than the other, bends the root toward moisture. No decision was made. The chemistry of differential cell expansion produced a directional outcome. Roots also detect and respond to the chemical signatures of decomposing organic matter, to the pH of the surrounding soil, to the metabolic byproducts of bacterial communities. When nitrogen is concentrated in a particular soil zone, roots entering that zone branch more prolifically, producing more lateral roots, more root hairs, more surface area to absorb what's available. When they leave that zone, branching slows. I find myself thinking about my foot on that root in Mount Airy, and how the root had no idea I was there. It had been growing across that trail for years, probably, following moisture and mineral gradients, completely indifferent to the path humans had worn above it. The trail existed because people kept walking it. The root existed because the chemistry of the soil beneath it kept pulling it forward. They intersected because geometry eventually makes everything intersect. There is a phenomenon called root foraging, which is studied seriously in plant biology, and which has an almost uncomfortably familiar quality when you read about it carefully. It turns out, don't invest equally across all soil zones they encounter. They perform something that looks functionally like cost-benefit analysis. When the carbon cost of extending a root into a new soil zone, the sugars required to build new root tissue, to maintain it, to support the respiration of its cells, exceeds the nutrient return that zone can provide. Root growth into that area slows or stops. When a zone is mineral rich, the opposite happens. Branching intensifies, root hair production increases, the tree commits more carbon to that location. The outcome is a root architecture that is, in a loose sense, shaped by the quality of the soil it has encountered. Not planned, not anticipated, built incrementally, each extension a response to the last reading of local conditions. When I finally stood up at Mount Airy, I looked back at the route for a moment. It crossed the trail in a long, low arc, vanished under leaf litter on one side, disappeared into the slope on the other. I had no way of knowing how far it went, how deep it dropped, what it had found or not found in the years since it first grew past this point. It looked completely unbothered, which of course it was. Roots of different species share soil space without merging. They detect each other. Root systems from neighbouring trees release chemical signals that influence the growth of roots in adjacent soil, sometimes inhibiting them, sometimes appearing to simply avoid each other through responses to shared chemical gradients. This is not cooperation in any deliberate sense, but the outcome is a kind of spatial organisation. Different root systems tend to occupy different depths and zones, reducing direct competition through the accumulated effect of individual growth responses. A forest floor, then, is underlaid by a root network that is organised, loosely, incompletely, always shifting, not through any plan but through the compounding of millions of small chemical responses over decades. The root that caught my foot was part of that, an edge of something vast and mostly invisible, following its own slow logic through the dark, occasionally breaking the surface in ways that surprise the people walking above it. There's something about the gap between what roots look like, static, fixed, just lying there across a trail, and what they actually are, actively growing, chemically sensing, continuously responding. That takes a moment to settle. You walk through a forest and the roots look like furniture, old, permanent, like they've always been exactly where they are. But the tip of every living root in that forest is moving right now, slowly, yes. A few millimeters a day, maybe less, but extending, sensing, branching or not branching depending on what the soil immediately ahead of it contains. The forest floor is quieter than the canopy, but underneath it something is always reaching, and sometimes, if you're walking without quite enough attention, it reaches you first. A fallen trunk is not an ending. It doesn't announce itself as one. It just comes down, in a storm, or slowly over years as the heartwood softens from the inside, or simply because the roots finally lost their argument with gravity, and then it stays, right where it landed, and the forest, without pausing, begins the long process of taking it apart. This process has no schedule, no urgency. It runs at the pace of organisms that measure their progress in millimeters per season, in enzyme concentrations, in the slow chemistry of wood that resists being undone. The first organisms to enter a fallen tree are rarely the most visible ones. Before the bracket fungi appear on the bark surface, before the beetle galleries are carved into the sapwood, something smaller has already begun. Bacteria colonize the moisture that collects in cracks and wounds. Fungal spores that have been drifting through the forest air for weeks land on the exposed wood and germinate if conditions allow, if the moisture content is right, if competing fungi haven't already established. Wood is mostly carbon, cellulose, hemicellulose, lignin, three structural polymers that gave the tree its rigidity while it was alive, and now present a formidable chemical problem for the organisms trying to disassemble them. Cellulose can be broken down by a reasonably wide range of fungi and bacteria. Hemicellulose, somewhat more difficult. Lignin is in a category of its own. Lignin is an irregular polymer, meaning its structure doesn't repeat in the orderly way that cellulose does. There's no single enzyme that can process it systematically. White rot fungi produce a suite of oxidative enzymes, lacases, peroxidases, that attack lignin through a process that is less like digestion and more like controlled damage. The enzymes generate reactive oxygen species that break chemical bonds somewhat indiscriminately, gradually fragmenting the lignin into smaller compounds that can then be processed further. It is slow. It takes years for white rot fungi to work through a large trunk, and not all of the lignin yields even then. Meanwhile, other organisms are arriving in sequence. Bark beetles detect volatile compounds released by the freshly fallen wood, terpenes and other chemicals that signal stressed or dead tissue, and bore through the bark to lay eggs in the cambium layer just beneath. Their larvae feed on the nutritious inner bark, carving galleries that branch and widen as they grow. These galleries do something structurally important. They open the interior of the log to moisture and air movement, creating the conditions that allow deeper fungal colonization. Beetle larvae are themselves food. Woodpeckers work fallen logs methodically. Predatory beetles follow the galleries of wood-boring species. The carbon that left the living tree as structural wood is being repackaged now into animal tissue, briefly before those animals die and return it to the soil or atmosphere in turn. What stands out most in this sequence is how each stage creates the conditions for the next one, not through coordination, through the blunt fact that feeding changes the substrate, and changed substrate attracts different organisms. The log that exists after 10 years of decomposition is not a degraded version of the original. It is a different habitat supporting a different community, running different chemistry. Carbon exits a decomposing log through several routes simultaneously. Fungal respiration releases carbon dioxide directly into the air inside and around the log. Leachate, water that has moved through the decomposing wood and dissolved organic compounds along the way, drains into the soil beneath the log, carrying carbon downward into the mineral layers. Fragments of partially decomposed wood are incorporated into the soil as invertebrates move through it, mixing organic material with mineral particles. And then there is biochar, not produced here but worth understanding in this context. When wood burns rather than decomposes, some of the carbon is converted into a highly stable aromatic form that resists microbial breakdown far more effectively than even lignin. A log that burns in a surface fire leaves behind charred material that may persist in the soil for centuries, while a log that decomposes slowly releases most of its carbon within decades. Fire and decomposition are, in a sense, competing pathways for the same carbon, one fast and partially stabilizing, one slow and more completely releasing. There is a quality that old decomposing logs have, a particular softness when you press your boot into one that's been down for many years, the way it yields without snapping, almost spongy with the work of fungi that have been running their chemistry through it for a decade or more. The wood has lost its structural integrity but hasn't disappeared. It holds moisture. It holds temperature. It has become something closer to soil than to wood, and the organisms living inside it are no longer the same ones that arrived in the first years. This is called coarse woody debris in ecological literature, and its carbon contribution to forest soils is significant enough that forests with active decomposing wood on their floors store meaningfully more soil carbon than forests where fallen logs are removed. The dead wood is not separate from the carbon budget of the living forest. It is still inside it, still cycling, still connected to the fungal networks that link into living root systems nearby. A large fallen trunk in an old-growth forest may take 80 or 100 years to fully incorporate into the soil. During that time, it will have housed dozens of species that nested or fed within it, contributed carbon to the soil in multiple chemical forms, altered the moisture and temperature regime of the ground beneath it, and eventually become indistinguishable from the surrounding duff layer. The tree that fell is gone in every visible sense long before that process completes, but its carbon is still here, redistributed, repackaged, held in fungal tissue and bacterial biomass and stable soil compounds, and the bodies of beetles that emerged from its galleries and flew to other parts of the forest. Nothing was lost, it only changed hands. There is something in the pace of this, the decades it takes, the unhurried sequence of organisms and chemistry that is worth sitting with for a moment before the next chapter. The forest doesn't rush decomposition. It doesn't need to. The carbon will move when the conditions allow it to move through the organisms equipped to move it, at the rate those organisms can work. The log lies where it fell, and around it and inside it, at a scale and pace below any casual observation, the work continues. It has been continuing since the day the tree came down, and it will continue long after the shape of the log has ceased to be recognizable from the surface. Light in a forest doesn't arrive evenly, it comes in pieces. A gap in the canopy lets through a column of it that moves across the understory floor as the sun shifts. A branch sways and a patch of leaf surface is suddenly exposed, then shaded again within seconds. The light environment in the lower layers of a forest is not dim in a uniform way. It is dim on average, but constantly interrupted by brief, irregular moments of higher intensity. These brief moments have a name, sunflecks, and the leaves down here have been shaped over evolutionary time to catch them. An understory leaf is built differently than a canopy leaf on the same tree species, or on a species that lives only in full sun. It tends to be thinner. The layers of cells responsible for photosynthesis, the palisade mesophyll, tightly packed columns of cells just beneath the upper surface, are less thick in shade-adapted leaves. There is less investment in photosynthetic machinery per unit of leaf area, because building and maintaining that machinery costs carbon, and carbon is exactly what a shaded leaf has less of to spend. The chloroplasts inside those cells are also doing something specific. In low light, they orient themselves face-on toward the incoming light, flattening themselves against the cell wall closest to the surface, maximizing the area intercepting whatever photons arrive. In high light, they rotate edge-on, reducing their exposure and protecting the photosynthetic machinery from damage. This reorientation is not fast by human standards. It takes minutes. But a sunfleck that lasts 30 seconds can still trigger a partial response. There is a threshold that every leaf operates near, in the understory. It's called the light compensation point, the light intensity at which the rate of photosynthesis exactly equals the rate of respiration. Below this threshold, the leaf is consuming more carbon than it's fixing, It's running a deficit. The tree is spending carbon to maintain a leaf that isn't earning its keep. Above the threshold, the leaf runs a surplus. Carbon fixed exceeds carbon spent. The leaf becomes a net contributor. In deep shade, many leaves spend significant portions of the day below their compensation point. What keeps them viable is the sun fleck, the brief exposure to higher light that pushes the rate of photosynthesis above respiration, if only for a short time, enough to tip the daily balance into positive. It is, when you think about it, an existence built around brief windows. Everything the understory leaf does is organized around capturing those moments when the light is suddenly, temporarily enough. You've probably experienced something like this in a different register, the way a conversation that had been going nowhere suddenly opens up around a single sentence, the way a long afternoon shifts when one thing finally resolves. The understory leaf isn't waiting in any conscious sense, but its entire architecture is an answer to the same problem. How do you sustain yourself in conditions that are almost but not quite sufficient? The answer the leaf arrived at over millions of years of selection is carefully, cheaply, with every structure pared down to the minimum required. Shade-adapted leaves often have higher concentrations of chlorophyll per unit of photosynthetic machinery than sun leaves do, not because they have more chloroplasts, but because each chloroplast contains more chlorophyll molecules, increasing the probability that any given photon will be absorbed rather than passing through unused. The pigment itself, chlorophyll, absorbs light primarily in the red and blue wavelengths, reflecting green. That reflected green is why leaves appear the color they do. The light the leaf can't use is the light you see. In the deep understory where most of the direct red and blue wavelengths have already been absorbed by the canopy above, the light that filters through is disproportionately green. Shade leaves have partially adapted to this. Some contain accessory pigments that can harvest wavelengths chlorophyll alone would miss. The machinery is extended incrementally to catch what the canopy left behind. There is something genuinely spare about all of this. No excess, no redundancy that isn't earning its carbon cost. The understory leaf exists at a kind of biological minimum, alive, functional, contributing what it can, in conditions that would shut down a sun-adapted leaf entirely. I find that more interesting than the canopy, honestly. The canopy is running at full capacity in good conditions. The understory is running near its limits all the time. The engineering required to stay viable under persistent near-insufficiency is, in some ways, more intricate than what's needed when resources are abundant. When a gap opens in the canopy, a large tree falls, a branch breaks, the light environment of the understory beneath it changes abruptly. Suddenly, the shade-adapted leaves are receiving far more light than they were built for. Their compensation systems, their chloroplast orientation responses, their limited enzymatic capacity, all of it is now being pushed past its design range. Some leaves bleach. The photosynthetic machinery is damaged by excess light in a process called photoinhibition. Reactive oxygen species accumulate faster than the leaf's protective systems can neutralize them, and the chlorophyll begins to break down. The gap is an opportunity, but it arrives faster than the existing leaves can adapt to it. What fills a gap eventually is new growth. Seedlings that have been waiting in suppressed states, sometimes for years, for exactly this change in light availability. They respond to the gap with rapid extension growth, investing carbon in height rather than leaf area, racing toward the light before the canopy closes again. The light that was never quite enough has suddenly become, briefly, more than enough. And then the canopy closes. New growth fills the gap over years. The understory dims again. The leaves that survived the transition, the ones that didn't bleach, that managed the sudden excess and then the return to shade, are still there, thinner than canopy leaves, oriented toward whatever light arrives, waiting in their non-waiting way for the next sunfleck to move across them. A few seconds of brighter light, the chloroplasts shifting face on, the brief arithmetic of enough. Somewhere below the canopy, a patch of light crosses leaf and moves on, and the leaf does what it has always done with whatever arrives. I'll be honest with you, my head is full of congestion and mild resentment right now, and the idea that I'm sitting here recording while my sinuses do whatever this is, it's fine. I've had worse nights, but there is something oddly fitting, and I didn't plan this, about talking tonight of all nights about things that leave and then quietly come back, because that's what a forest does, chemically, above its own canopy. It exhales, continuously, invisibly, through every leaf versible surface and every bark pore and every gap in the canopy where warm air rises. Volatile compounds drift upward, terpenes, isoprene, a range of smaller organic molecules that forests produce as byproducts of their own internal chemistry. Some of these compounds have smell. If you've ever stood in a pine forest in midsummer heat and noticed something sharp and clean in the air, something that doesn't belong to any single thing you can point to, that's partly this. Terpenes off-gassing from resin, from leaf surfaces, from the duff layer warming in the afternoon. The forest is always doing this. You just don't always notice. These volatile organic compounds, VOCs in the shorthand, don't simply disappear into the atmosphere and keep going forever. They react. Once airborne, they encounter hydroxyl radicals and ozone, and they oxidize. The products of these reactions are less volatile, stickier, more likely to condense onto particles or surfaces. Some of them aggregate into tiny aerosol particles that scatter light. This is part of why distant forested mountains sometimes have a faint blue haze above them. The Great Smoky Mountains are named for it. That haze is, in part, forest carbon that left through leaf pores and then changed form above the canopy. Some of these particles settle back down, onto leaf surfaces, onto soil, into streams running through the forest. Some are incorporated into cloud droplets and return in rainfall, dissolved organic carbon arriving from above, entering the soil, potentially taken up by roots. The carbon that left through a stomate on a Tuesday might return, chemically altered, as part of a raindrop weeks later. I keep thinking about this and then losing the thread because my brain is operating at maybe 60% tonight. The general point is that the forest's carbon boundary is not its canopy. The chemical influence of a forest extends upward into the atmosphere above it, in ways that feed back down. It doesn't stay gone. The atmosphere directly above a dense forest canopy is chemically distinct from the air a few kilometers away from the tree line. Higher concentrations of VOCs, higher particle counts, different humidity profiles because of all the water vapor the canopy is releasing. The forest is, in a real sense, generating its own local atmospheric conditions. This matters for precipitation. Aerosol particles act as condensation nuclei, surfaces onto which water vapor can condense to form cloud droplets. Forests in some regions appear to influence their own rainfall, at least locally and partially, by seeding the atmosphere above them with the particles their own chemistry produces. The forest exhales. The exhalation seeds clouds. The clouds rain. The rain returns carbon and water to the forest. It's a loop, imperfect, variable, not operating on any tidy schedule, but running. You know what doesn't loop? A cold. A cold just sits there, doing its thing. Oh, elegant return to baseline, just several days of your body being extremely committed to a project you didn't agree to. Anyway, isoprene is worth a moment specifically because it's produced in quantities large enough to matter at global scales. Trees, particularly oaks, poplars, and some tropical species, emit isoprene at rates that increase with temperature and light intensity. On a hot summer day, a large oak may emit several milligrams of isoprene per gram of dry leaf weight per hour. Across a forest, across a continent, this adds up to something the atmosphere actually has to account for. Why plants produce isoprene is still genuinely not fully resolved. The leading hypothesis is that it provides thermal protection. Isoprene appears to stabilize membrane lipids in leaf cells under heat stress, reducing photoinhibition damage during temperature spikes. The emission may be a byproduct of this protective function, rather than something produced for the purpose of entering the atmosphere. The forest isn't trying to seed clouds, it's trying to keep its leaves from overheating on a hot afternoon. The atmospheric consequences are incidental. Biology is full of this, consequences that dwarf the intention. There is also something happening at the forest floor that connects to all of this, and it's easy to miss because it doesn't have a dramatic visual. When rain carries dissolved organic carbon into the soil, some of it is immediately available to soil microbes. Some binds to mineral surfaces and stabilizes, as discussed earlier, and some is taken up by roots, not as a primary carbon source since the tree is producing its own carbon through photosynthesis, but as supplemental input, particularly for certain nitrogen-containing compounds that arrive dissolved in rainfall and can be absorbed directly. The carbon that went up as a volatile compound, oxidized in the atmosphere, returned in rain, entered the soil, and was taken up by a root hair. That carbon has travelled. Not far by any geological standard, but it left the tree, changed form twice, and came back. Honestly, the more I sit with this, the more I think forests are just very good at not losing things, not perfectly. Carbon does leave permanently, carried away in stream water, buried in sediment, exhaled into an atmosphere that disperses it globally. Losses happen, the cycle is real, but the tendency, if you watch it at the right scale, is toward return, toward recapture. The forest keeps pulling things back in, not through intention but through the sheer density of the chemistry it maintains in the air and soil and water immediately around itself. The pine smell that hits you when you walk into a warm conifer forest. That's not decoration, that's the forest's chemistry, briefly entering your lungs, your bloodstream, your olfactory receptors. Carbon that was inside a cell an hour ago, inside you now, exhaled back into the air in a few seconds. You were briefly part of the loop too, which is either a beautiful thought or a strange one depending on how your sinuses are doing. The carbon doesn't stay gone. It finds its way back to something eventually in a form that something else can use. There's a version of letting go that isn't dramatic, not the kind you have to talk yourself into, not the kind that requires a decision, just the slow biochemical realization that something has already started leaving, and the most useful thing you can do now is take back what you can before it goes completely. Trees do this every autumn, and they were remarkably unsentimental about it. Somewhere around late summer, before the air temperature drops enough for you to notice the shift, before the first leaf has visibly changed colour, the tree has already started. Day length, that's the trigger. Not cold, not necessarily. Photoperiod, the ratio of light hours to dark hours in a 24-hour cycle, is detected by photoreceptors in the leaf tissue, and when the dark period crosses a species-specific threshold, a hormonal signal begins moving through the tree. Oxine levels in the leaf drop. Ethylene increases. A layer of cells at the base of each leaf stalk, the abscission zone, begins to differentiate. The cells there stop dividing and start changing their wall chemistry, producing enzymes that will eventually dissolve the connections between them. The leaf doesn't know it's being prepared for release. There's no knowing involved. The cells are just responding to a local hormonal environment that has shifted, doing what their gene expression now dictates. But before that abscission is complete, before the leaf falls, the tree does something that I think gets undersold in most descriptions of autumn. It retrieves. Chlorophyll breaks down. This is what reveals the yellows and oranges that were always present in the leaf but masked all summer by the sheer density of green pigment. The nitrogen locked inside chlorophyll molecules is actively dismantled and exported back through the phloem into the woody tissue of the twig and branch. Phosphorus follows. Certain metal ions, some carbon compounds. The leaf is being systematically emptied of what the tree has decided is worth keeping. You know that feeling when you're leaving a place, a house, a city, a version of your life, and you're packing, and at some point you stop asking, should I keep this? And start asking, is this actually worth carrying? The tree is doing exactly that, not metaphorically. The tree is running a literal cost-benefit calculation on every molecule in that leaf tissue, moving through vascular channels, deciding what crosses the abscission zone before it seals. What doesn't make it back? Structural carbon in cell walls, residual compounds in the mesophyll, pigments that broke down too late to be reabsorbed, leaves with the leaf. The forest floor receives what the canopy decided wasn't worth the transport cost. Nitrogen resorption efficiency, the percentage of leaf nitrogen a tree manages to recover before leaf drop, varies by species, by soil nitrogen availability, by how much time the tree has before conditions force abscission. Deciduous trees in nitrogen-poor soils tend to resorb more efficiently than those in nitrogen-rich soils. When nitrogen is hard to come by in the ground, the tree works harder to recover what it already has invested in its leaves. Scarcity sharpens the accounting. It always does, doesn't it? Scarcity has a way of making you very precise about what actually matters. The colour change isn't just chlorophyll leaving. Some trees, maples notably, actively synthesize new pigments in the leaf during senescence. Anthocyanins, which produce reds and purples, are produced from sugars that accumulate in the leaf as the phloem connection to the rest of the tree begins to close. The leaf is filling with the sugar it can no longer export, and it converts some of it into pigment. Why? Genuinely not fully settled. The leading hypotheses involve photoprotection. The anthocyanins may shield the leaf's recovering machinery from excess light while resorption is still underway, or signalling to insects, particularly aphids, which apparently prefer to lay eggs on trees with less anthocyanin. Brighter red may mean the tree has more resources to invest in chemical defense. A brilliant red maple in October might be, among other things, a warning, or a sunscreen, possibly both. There's something I find quietly moving about the timing of all this, and I don't say that in a vague, poetic way. I mean something specific. The tree starts preparing for leaf drop before any external signal that a human observer would register as autumn. The internal chemistry is already shifting in late July, in early August, while the air still feels like summer and the leaves still look fully green and operational. The senescence program has already been initiated. The retrieval has already begun. You're looking at a leaf that is already, in some cellular sense, on its way out, and it looks fine. It looks like it's doing exactly what it's always been doing. How many things are already in the process of ending, while still appearing entirely continuous? Not a dark thought, just a true one. After abscission, the leaf falls. Where it lands matters. A leaf that falls onto the soil surface beneath its own tree enters decomposition. Fungi begin processing the remaining carbon, bacteria colonize the softening tissue, invertebrates fragment it physically, and over months the leaf becomes something the soil can absorb. Some of that carbon, the part that doesn't volatilize as carbon dioxide during decomposition, enters the mineral soil. Some will be taken up by roots next spring. Some will bind to clay particles and sit there for a long time. The nitrogen and phosphorus that the tree retrieved in October will be remobilized in March, pushed into new buds, built into the leaves of next season. The carbon that left with the fallen leaf might return through the roots within a year, so the leaf falls, and the tree stands there, bare, branches holding whatever light the winter sun offers, running on stored reserves, waiting for the photoperiod to shift back. It has done this before, many times. Every time it has retrieved what it could, released what it had to, and waited. There's a patience in that which isn't stoicism, it's not a decision. But from the outside it looks like something, like a kind of competence with impermanence, like knowing at some deep chemical level that what leaves is not entirely lost. Some of it comes back. It always has. The tree stands in the cold and holds that knowledge in its chemistry, in its stored nitrogen, in the abscission scars where the leaves were. Small pale marks on every twig, proof that something was here and was released, and that the tree is still, despite all of it, intact. Drought is not a dramatic thing when it starts. It doesn't arrive with a sound, no announcement. One week the soil is adequately moist, the next week it's a little less so, then a little less than that. And the forest, which has no weather app, no forecast, no concept of the word drought, begins responding to conditions it can only measure through chemistry, through turgor pressure in cells, through the concentration of solutes in the water. It's managing to pull from soil that is slowly, quietly tightening its grip. I think about this sometimes when I'm mildly dehydrated and still somehow refusing to drink water. The forest and I have more in common than I'd like to admit. When water becomes scarce, a tree's stomata begin to close. This is a protective response. Closing the stomata reduces water loss through transpiration, but it also reduces carbon dioxide intake. Less CO2 means slower photosynthesis. Slower photosynthesis means less sugar produced. Less sugar means less carbon available to send downward through the phloem. The mycorrhizal fungi attached to the roots notice this, not consciously, but the carbon flow they depend on slows, and their physiology responds to that slowdown the way any organism responds to a reduction in food supply, by pulling back, growing less, allocating less energy to maintaining existing hyphal connections. Some connections hold, some don't. Here's the part that I find genuinely uncomfortable to think about, in that specific way where something true is also a little bleak. The fungi that survive drought best, the ones that maintain their connection to the tree through extended dry periods, tend to be species that are either more drought tolerant in their own right, or species that have a longer established relationship with that particular host. Newer connections, shallower relationships, partnerships that hadn't fully ramified through the soil, these tend to break first. It's not personal, it's hydraulics and carbon budgets, but the outcome is that drought selects for persistence. The network that exists after a prolonged dry period is composed of the partnerships that were either deepest or toughest. Everything more provisional got pruned by circumstance. I've met people who work this way too. You find out who actually stays when things get genuinely inconvenient. The forest has apparently always known this. The tree, meanwhile, is doing something specific with the water it has left. Abscisic acid, ABA, is produced in the roots when soil moisture drops below a threshold and is transported upward through the xylem to the leaves, where it triggers stomatal closure. This is one of the more elegant long-distance signaling systems in plant biology. The root detects the problem, synthesizes a hormone, ships it upward in the water stream, and the leaf responds before it has directly experienced the water deficit itself. The leaf closes its stomata based on a message from the root. The root sent that message based on what the soil around it felt like. There is no brain in this system. There is no central processor. Just chemistry moving through tissue, each component responding to local conditions, the whole thing producing coordinated behavior across an organism that might be 30 meters tall. I have a spinal cord and a prefrontal cortex, and I still handle stress less elegantly than this. Make of that what you will. Now, the mycorrhizal network under these conditions. The network has thinned. Some fungal species have retreated, their hyphae have died back in the driest zones, and the phosphorus transport those hyphae provided has dropped proportionally. The tree is receiving less mineral support at exactly the moment it needs to be running efficiently. But here is the thing that doesn't get enough attention. Some ectomycorrhizal fungi, particularly species that form thick hyphal sheaths around root tips rather than penetrating between root cells, appear to provide a hydraulic function during drought. Their hyphal networks can move water from moister soil zones toward the root surface, functioning as a supplemental water delivery system when the soil immediately surrounding the root has dried out. The fungus, in other words, can extend the tree's effective reach into the soil moisture gradient. Not dramatically, not enough to negate a severe drought, but enough to matter at the margins, which in biology is often where survival is decided. There's a particular kind of friend, and I think you know this person, who is absolutely useless in normal circumstances. Bad at texting back, forgets plans, generally chaotic. And then something actually goes wrong and they are suddenly inexplicably exactly what you needed. Show up with food, stay the right amount of time, say the one correct thing. Some mycorrhizal partnerships feel like that to me. Minor contributors during good conditions, quietly essential when things get hard. After the drought breaks, when rainfall returns, when soil moisture climbs back up, the tree resumes fuller photosynthesis. Carbon flow downward increases. The surviving fungal partners begin expanding again, producing new hyphae, recolonizing soil zones that dried out during the drought. But the network that reconstitutes is not the same one that existed before. The species composition has shifted. Drought-sensitive fungi that were present before may not re-establish quickly, or at all. New colonizers may arrive from spores in the soil that belong to species better adapted to the drier conditions the forest is now experiencing more frequently. The partnership reassembles, but with different partners in different proportions. The forest doesn't grieve this. It can't. It just runs the next version of the network with whatever is available. Which brings me to the part I think about most, which is what repeated drought cycles do over decades. Each drought episode selects the network toward greater drought tolerance. Species that can't handle dry conditions are progressively underrepresented. The network that exists after 5 drought cycles is not the same network that would have developed under stable moisture conditions. This is adaptation. Real, observable, documented adaptation. Not of the tree itself, but of the community of organisms the tree depends on. The forest is being shaped by stress in ways that extend below the soil surface, into the composition of relationships it maintains with organisms it can't see. The tree looks the same from the outside, the network underneath it is different. It usually is, after enough hard seasons. Tonight the soil somewhere is drier than it was last month. The fungal threads in that soil are thinner than they were in spring. The tree above them is moving a little less carbon downward than it did in June. Nothing about this is visible. Nothing about it sounds like anything. It is happening in complete silence, in complete darkness, in a medium most people will never willingly touch. And it has been happening, this exact negotiation between tree and fungus and soil moisture, for 450 million years, through droughts that made this one look mild. The network thins, the toughest threads hold, and the forest waits for rain with the particular patience of something that has waited before and knows, through no knowing at all, that waiting works. Fire gets a bad reputation, and look, fair enough, it's destructive, visually terrifying. Culturally, we've spent millennia treating fire as the thing that ends things. In basically every mythology I can think of, fire is either divine punishment or the precursor to rebirth, and both framings agree on one thing. Fire is a before and after event, the world before the fire, the world after, but the biology doesn't quite work that way. When fire moves through a forest, not a catastrophic crown fire that incinerates everything, but a surface fire, the kind that burns through the understory and leaf litter and downed wood, it moves fast enough that the soil beneath it barely registers the temperature change below the first few centimeters. The organisms living deeper in the soil survive. The seed bank, seeds buried in the mineral soil, often survives. What burns is the surface, the accumulated organic matter, the duff, the fallen branches, the shrub layer, and as that material burns, its carbon exits rapidly as carbon dioxide. Years of slow decomposition compressed into hours, the carbon that a fallen log was releasing incrementally over decades, gone in an afternoon. That part is real loss, genuinely. I'm not going to romanticize it into something it isn't. But not all of the burning material converts cleanly to carbon dioxide. Some of it, particularly woody material that burns incompletely in low oxygen conditions at the edges of the fire, converts to charred carbon, biochar, a structurally altered form of carbon that is, chemically, almost aggressively stable. Biochar is mostly graphitic carbon, carbon atoms arranged in ring structures that resist microbial enzyme systems. The bonds holding it together don't offer the same chemical handholds that cellulose or lignin do. Most decomposers simply cannot get purchase on it, so it sits. In the soil, sometimes for thousands of years, in forms that can be dated and identified in soil cores from forests that haven't burned in recorded human memory. The fire happened, the carbon was altered, and then it stayed, in that altered form, long after every other trace of the fire had disappeared. This is the part I find almost unreasonably interesting, and I'll tell you why. We think of burning as the ultimate destruction. You burn something, it's gone. That's the whole cultural weight of the image, the irreversibility of it, the completeness of it. And yes, the volatile fraction of the wood does leave permanently. But a portion of what burned is now more stable than it was before the fire. The fire didn't destroy that carbon, it changed it into something harder to destroy. There's a version of this in human experience that I don't want to overstate, but also, it's there. The things that have been through serious heat sometimes come out in a form that is less flexible, less lush, less immediately useful, but also less likely to decompose under ordinary conditions, less available to the usual processes that break things down. I'm not saying trauma is good, I'm saying the analogy exists and I noticed it and now you have to think about it too. Post-fire soils are strange. The ash layer on the surface is alkaline. Wood ash contains calcium, potassium, magnesium released from the organic matter that held them. These minerals are now immediately available at the soil surface, in concentrations that can temporarily spike nutrient availability to levels the soil rarely sees under normal conditions. Plants that establish in post-fire environments often experience a brief flush of growth from this nutrient pulse. Then the pulse fades as the minerals leach downward with rainfall or are taken up by the first colonizing vegetation. The window is narrow, but the biochar remains. And biochar does something the alkaline ash doesn't. It persists as a long-term soil amendment, creating pore structures that hold water and provide habitat for microbial communities. Soils with significant biochar content tend to have different microbial community compositions than adjacent unburned soils, different water retention characteristics, different long-term carbon dynamics. The fire changed the soil's operating conditions for decades after the smoke cleared. There are also fire-adapted species, plants whose reproduction is either triggered by or dependent on fire passing through. Lodgepole pines produce serotinous cones sealed with resin that melts in the heat of a fire, releasing the seeds inside. The cone can persist on the tree for years, decades even, waiting. Fire comes, resin melts, seeds fall onto a landscape that is suddenly mineral-rich, canopy open, competition cleared. The cone was built around the assumption that fire would come. Its entire reproductive strategy is contingent on destruction. That's either bleak or elegant depending on your current mood, and frankly I've felt both about it at different times. What fire returns then is complicated. It returns minerals to the surface in immediately available form. It returns carbon to the atmosphere rapidly in a pulse that the slow process of decomposition would have spread across decades. It returns light to the forest floor by clearing the canopy and understory. And it converts a fraction of the carbon it processes into a form that returns to the soil in a condition that will outlast almost everything else in it. Some of what fire takes it gives back differently. Some of what it takes it gives back in a form that lasts longer than what was there before. The soil carbon record in fire-prone forests is a kind of archive of every burn that has passed through, going back further than any human observer recorded. Each fire left a horizon of charred material, a dark layer in the soil profile, sometimes thin, sometimes thick depending on intensity and fuel load, datable by the radiocarbon in the charred organic matter. The forest carries its fire history in its soil, the way a person carries certain things in their body, not visibly, not consciously, but structurally, in the way things have been arranged by what passed through. And here's the part that I keep sitting with. A forest that has never burned accumulates organic matter at the surface, duff, fallen wood, undecomposed leaf litter, that eventually becomes a significant fuel load. The longer the fire is absent, the more material there is to burn when fire finally comes, and the more intense that fire will be when it arrives. Paradoxically, suppressing fire doesn't remove fire from the ecosystem, it defers it, and it accumulates the conditions that make the eventual fire worse. Some ecologists argue that low-intensity, frequent fire, the kind that many fire-adapted ecosystems evolved with, is less destructive over the long run than infrequent, high-intensity fire, that the attempt to prevent fire entirely has, in many landscapes, produced the conditions for exactly the catastrophic fires the suppression was meant to avoid. The forest apparently would have preferred we let it burn a little regularly, rather than protecting it perfectly until it couldn't be protected at all. I feel like there's a parenting philosophy somewhere in there, but I'll leave that alone. Tonight, somewhere, there is a soil with charred carbon in it from a fire that burned before anyone currently alive was born. The biochar in that layer has been sitting there, chemically stable, while the forest above it went through drought and regrowth and succession and the whole long rotation of the carbon cycle. The fire that made it is gone. Everything that burned in that fire is long since dispersed. But the carbon it converted, that particular fraction, the incompletely burned fraction, is still there. Still there. It turned out to be the most durable thing the fire produced. There is a particular kind of presence that dead things have in a forest. Not eerie, not heavy, just present. A standing dead tree, what ecologists call a snag, occupies space the way living trees do. It blocks light. It holds moisture. Birds use it. Insects colonize it. Rain hits it and runs down its bark and into the soil at its base the same way it always did. From a distance in the right season, you might not immediately register that it's dead at all, and its carbon, all of it still there, is just beginning a different kind of accounting. I want to be upfront about something. We talk about death in biology with this very clean narrative. The organism dies, decomposition begins, carbon returns to the cycle. It's tidy. Educational diagrams love it. A neat arrow from dead organism to decomposers to soil to atmosphere to plant and back again. What actually happens is messier, slower, and honestly more interesting than any diagram I've ever seen has bothered to communicate. When a tree dies standing, or as opposed to falling immediately, it enters a decomposition sequence that is fundamentally different from a fallen log. The bark, still intact, creates a different moisture regime. The wood inside is still vertically oriented, which affects how water moves through it, how fungi colonize it, how the whole thing structurally fails over time. Fungi enter through wounds, old branch scars, broken tops, places where the bark has already been compromised. They begin working inward, softening the heartwood first, the dense, darker inner wood that in a living tree was already non-conducting, already largely composed of lignified dead cells. The heartwood is structurally critical but metabolically inert. It's the tree's skeleton, and it is, accordingly, the hardest part to decompose. Lignin again, always lignin. The heartwood of a large tree is among the most lignin-dense material in a forest. White rot fungi are the primary organisms capable of meaningfully processing it, and they work at a pace that would frustrate anyone operating on human timescales. A large snag, say a Douglas fir that died at 200 years old with a trunk diameter of a metre, may stand for 50 or 60 years before structural failure brings it down. During that time, the internal decomposition is working outward from wherever the fungi entered, but the outer shell of sapwood and bark may remain surprisingly intact. The tree looks dead. The interior is being slowly hollowed. Here's a niche analogy that I can't help making. There's a technique in traditional Japanese joinery, shosugiban, where cedar is charred on the surface to create a protective layer. The burning paradoxically preserves the wood beneath it. A dead tree in late-stage decay is doing something structurally similar, but in reverse. The exterior holding form while the interior transforms. The shell persists longest. The core goes first. I have no idea if that's meaningful or just two things that superficially resemble each other, but I noticed it, and here we are. The carbon leaving a standing dead tree doesn't leave all at once, or even close to it. Fungal respiration releases carbon dioxide steadily but slowly. Bark beetles and wood-boring insects create galleries that accelerate moisture infiltration and fungal spread, but their contribution to direct carbon release is minor. What they mostly do is open the wood to organisms that will release carbon later. Woodpeckers excavating nest cavities in snags are, without intending to, restructuring the decomposition timeline of a carbon store that might otherwise have remained sealed for additional decades. Every organism that interacts with the dead tree is, in some functional sense, participating in a carbon release event that will play out across a century. Here is the opinion part clearly labelled as such. I think the cultural instinct to clean up dead trees, to remove snags from forests, to clear fallen wood, to tidy the places where things have died, is one of the more quietly costly aesthetic preferences we have ever acted on at scale. Managed forests with active snag removal store less carbon. They support fewer cavity nesting species. They have thinner soil organic layers. The tidiness costs something real, and the cost is paid by organisms that don't get a vote in the decision. A forest with dead wood in it looks, to a certain kind of eye, neglected. To a different eye, one that has spent any time understanding what's happening in that wood, it looks functional. Alive in a different register. I know which eye I prefer. I'm biased. I'm telling you I'm biased. There is a species of beetle, the California forest borer, among others, whose larvae require dead or dying conifers at specific stages of decay. Not freshly dead, not fully decomposed. A very specific window of moisture content and fungal colonization that occurs somewhere in the middle years of a snag's decomposition. Outside that window, the larvae cannot develop. The beetle's entire existence depends on the forest maintaining dead wood across a range of decay stages simultaneously. A forest with snags of different ages and species and decay states supports the beetle. A forest where dead wood is systematically removed, or where all the snags are the same age, perhaps because a single fire or logging event created them all at once, cannot. Biodiversity in this context is partly a function of temporal diversity. The forest needs to be dying at different rates in different places, continuously to support the full community of organisms that depend on specific stages of dying. Death needs to be distributed, unscheduled, ongoing. The carbon in a dead tree, then, is not simply waiting to be released. It is actively supporting a community of organisms whose existence depends on the particular chemistry of decomposition at each stage. The carbon that feeds the beetle larva becomes the beetle, which is eaten by a woodpecker, which excavates a cavity that is later used by a flying squirrel, which is eaten by an owl, which dies somewhere else in the forest and contributes its carbon to a completely different patch of soil. The dead tree's carbon budget has branches the tree never grew. Eventually the snag falls. Structurally weakened by decades of internal decomposition, it comes down in a storm or simply under its own weight, and the above-ground decomposition sequence continues on the ground. The same processes, different geometry, different moisture dynamics, different community of organisms moving in. A large tree that died standing might spend 50 years as a snag and then another 100 as a fallen log before its carbon is fully incorporated into the soil. 150 years from death to soil. And even then, even after the wood is unrecognizable, even after the carbon has moved through fungal bodies and bacterial biomass and soil organic matter, some fraction of it has reached the mineral-associated layer, bound to clay, stabilized. That fraction isn't cycling anymore. It arrived at a kind of rest that the living tree, with all its continuous metabolic activity, never got to experience. The forest where that tree spent 200 years growing is different now. Different light reaches the ground where the crown used to be. Different species are establishing in the gap. Different chemistry is running through the soil where the roots are slowly releasing their own carbon back into the ground. The tree is gone in every way that matters to a casual observer. But in the soil, in the organisms that move through it, in the cavity that was excavated in its trunk 20 years before it fell and is now a collapsed hollow in the duff, in all of these places the tree is still, in some distributed sense, present. The accounting isn't finished yet. It won't be for a long time. Something changes when the light goes, not dramatically. The forest doesn't hold its breath at dusk and release it at dawn. It doesn't pause, but the chemistry shifts, quietly, completely. And the forest that exists at 2 a.m. is running a fundamentally different carbon equation than the one that existed at 2 p.m. You probably already sense this, even if you've never thought about it in biological terms. Forests feel different at night. The air is different. The smell is different. Something about the quality of the darkness in a forest is distinct from the darkness of an open field, and part of that difference is chemical. The forest is exhaling continuously into the still air trapped beneath the canopy. During the day, photosynthesis and respiration run simultaneously. Every living cell in the forest and every root tip, every fungal hypha, every bacterium in the soil, every cell in every leaf is respiring continuously, consuming oxygen and releasing carbon dioxide. This is cellular respiration, and it doesn't stop for light or dark or season. It is the baseline metabolic hum of everything alive. During daylight, the leaves add photosynthesis on top of that. Carbon dioxide is being pulled in through the stomata, fixed into sugars, incorporated into the tree's growing carbon pool. In good light, a healthy leaf fixes carbon at a rate that exceeds what it releases through respiration, and the excess is exported, moved through the phloem, distributed to wherever the tree needs it. The forest on a sunny day with adequate water is a net carbon sink, taking in more than it releases. At night, photosynthesis stops entirely. The stomata close, not completely, in all species but largely. The leaf is no longer fixing carbon, but every cell in the leaf is still respiring, still consuming, still releasing carbon dioxide into the intercellular spaces inside the leaf, which slowly diffuses outward through the partially closed stomata and the thin cuticle. The forest's carbon balance in darkness tips toward release. I recorded most of this chapter to just after midnight. My cold has settled into that specific phase where you feel almost normal until you try to breathe through your nose, at which point reality reasserts itself. The house is quiet. The cooling system just kicked on. There's that low hum that you stop hearing after 30 seconds, but you notice when it starts. I've got tea, the kind that promises things it can't deliver. And I keep thinking about the fact that outside, right now, every forest is doing this, running its nighttime carbon math quietly without anyone watching. The roots are perhaps the most active component of the forest's nighttime respiration. Root respiration is continuous and substantial. In many forest ecosystems, roots and their associated mycorrhizal fungi account for roughly half of the total soil carbon dioxide flux. This isn't something that varies much with light, because roots don't photosynthesize. They respire around the clock, consuming carbohydrates that the leaves produce during the day and sent downward through the phloem. At night, that downward flow of carbohydrates continues. The phloem doesn't shut off with the light. Sugars produced during the day move toward the roots through the night, fueling the root growth and maintenance and mycorrhizal exchange that the tree's daytime photosynthesis made possible. The day's light is still doing work hours after it ended. Soil respiration at night has a particular quality that I find worth sitting with. The carbon dioxide released by roots and soil microbes doesn't immediately diffuse up through the soil and into the atmosphere. It moves slowly through the soil pore space, dissolving into soil water, pooling in soil layers where diffusion is slow. Dense clay layers, compacted zones, the boundary between organic and mineral horizons. Carbon dioxide concentrations inside the soil can be 10, 20, sometimes 50 times higher than atmospheric concentrations. This pool of dissolved carbon dioxide in soil water, carbonic acid effectively, is part of the chemistry that weathers minerals over time, slowly releasing calcium and magnesium from rock surfaces. The forest's nighttime respiration is incrementally dissolving the geology beneath it. That is either a magnificent or a deeply unsettling thought, depending on your mood. The forest floor is also doing something specific at night that it doesn't do during the day. Dew formation, condensation of water vapor onto leaf surfaces, onto soil particles, onto the surfaces of decomposing wood, adds moisture to the system during the night that wasn't there at dusk. This moisture pulse is minor compared to rainfall, but it is continuous and reliable in many forest environments. The organisms that decompose leaf litter and wood near the surface benefit from this nightly rehydration. Fungal hyphae, that might have dried slightly during the afternoon extend into the dew-moistened surface. Microbial activity in the upper millimeters of soil increases. The night provides water, quietly, without drama. There is a concept in forest ecology called nocturnal carbon balance. The accounting of how much carbon the forest releases during the dark hours versus how much it fixed during the light hours. Forests in which the nighttime release approaches or exceeds the daytime fixation are, over a full day, nearly carbon neutral or even slight carbon sources. This tends to happen during periods of heat stress. High nighttime temperatures increase respiration rates, while high daytime temperatures can inhibit photosynthesis by causing stomatal closure to prevent water loss. A forest can have a green, apparently healthy canopy and still be releasing more carbon than it fixes if the temperatures are high enough at night. This is not a hypothetical concern, it is being measured. The numbers are not always comfortable. I want to say something here that I think is true, but that I hold with some uncertainty. The forest at night feels to me more honest than the forest during the day. During the day, the photosynthesis is running, the canopy is fixing carbon, the whole system looks productive and self-sustaining, and from the outside, at night, you get to see what the baseline actually is. The respiration that was always there, now uncompensated. The carbon release that was always running beneath the fixation, now visible in the net balance. The forest doesn't perform at night, it just runs. And what it runs on in the dark is what was stored in the light. There's something in that about the relationship between effort and rest that I don't want to make too neat. Biology doesn't offer clean lessons, but the architecture of it, the way the nighttime depends entirely on what the daytime built, the way the roots are fed through the dark by sugars the leaves made in the sun, it's not nothing. The darkness is not empty. It's full of everything that the light made possible. By morning, the carbon dioxide concentration in the air beneath the forest canopy is measurably higher than it was at dusk. The night's respiration has enriched the air close to the ground. When the light returns and the stomata open and photosynthesis resumes, the leaves are opening into air that is slightly carbon-rich, a small advantage, a minor head start. The night produced that. The exhalation of roots and soil and every living cell in the forest, running through the dark hours, left something behind in the air that the morning can use. The hum of the cooling system just stopped. The house is quieter now, and somewhere outside, in whatever green space exists beyond these walls, the same chemistry is running that has been running in forests since before there were forests as we currently define them, since the first land plants began respiring in the dark of whatever nights existed 400 million years ago. Continuous, patient, unobserved, the forest breathes at night because it has no other option and because everything it will do tomorrow depends on not stopping. Water doesn't want to go up. That's not a poetic observation, it's physics. Gravity pulls water down and moving it upward requires energy, requires something to do the work of lifting it against that pull. And yet, in a tree 30 meters tall, water is moving upward continuously, from root hair to leaf surface, through a column that in some species reaches over 100 meters without a single pump anywhere in the system. No heart, no peristalsis, no mechanical pump of any kind, just physics, applied very carefully. The mechanism is called the cohesion tension theory, and it is, I think, one of the most underappreciated pieces of plant biology in existence. Not because it's obscure, it's in every introductory textbook, but because the textbook version tends to flatten it into a clean explanation that loses the genuinely strange part. Here is the strange part. The water column inside the xylem, the network of hollow, dead cells that forms the tree's water transport system, is under negative pressure. Tension. The water is being pulled upward, not pushed, and the force doing the pulling is evaporation at the leaf surface. When water evaporates from the cell walls inside a leaf, moving out through the stomata as water vapor, it creates a slight deficit of water in those cells. That deficit pulls water from adjacent cells, which pulls from the cells adjacent to those, which pulls from the xylem vessels in the leaf, which pulls from the xylem in the branch, which pulls from the trunk, which pulls from the roots. The entire column of water from root to leaf is connected, cohesive, because water molecules are strongly attracted to each other through hydrogen bonding, and the evaporation at the top is the engine that pulls the whole column upward. The tree is drinking through its leaves. Okay, here's my niche analogy and I'm committing to it. You know those old school vacuum tube audio amplifiers? The kind that audiophiles spend unreasonable amounts of money on because they claim the sound is warmer, and the whole circuit depends on maintaining a very specific electron flow through a tube under carefully controlled pressure conditions, and if one thing in the system goes slightly wrong, the whole signal degrades. Xylem water transport is like that. The tension in the water column has to be maintained within limits. If air enters the column, if a bubble forms anywhere in the xylem, the tension breaks locally. The water column in that vessel cavitates. That vessel stops conducting. The tree has strategies to manage this. It has many xylem vessels, so losing some doesn't stop the whole system. It can refill cavitated vessels under certain conditions, but the vulnerability is real, and under drought stress, when the tension in the water column increases because the soil is dry and the leaves are still evaporating, cavitation events increase. The signal degrades. The system loses conductance. Carbon moves in the opposite direction. This is the thing that I think gets glossed over in diagrams that show both xylem and phloem, without quite conveying the elegance of the arrangement. Two parallel transport systems running through every branch and trunk and twig, one pulling water and dissolved minerals upward, one moving sugars and other organic compounds downward, both running simultaneously in tissues that are only millimeters apart. Phloem transport is driven by pressure differences. Sugars are loaded into phloem cells at sites of production, the leaves, which increases solute concentration, which draws water in osmotically, which creates pressure. At the other end, where sugars are consumed, roots, growing meristems, developing fruit, solutes are removed, water leaves, pressure drops. The pressure gradient between source and sink drives the flow. The phloem is, in a loose sense, pressurized from the top. The xylem is pulled from the top. Both systems are driven by what the leaves do. The leaf is not just a photosynthetic organ, it is the engine of the entire transport system of the tree. I've been thinking about this chapter all day, which, given that my cold has me firmly in the mildly miserable but functional zone, is saying something. There's something about the xylem that I keep returning to, the idea that the whole system runs on tension, that the water is being held in a state of continuous stress, and that the tree's ability to function depends on maintaining that stress within survivable limits. That metaphor writes itself, and I'm trying not to let it, but I'd be lying if I said I didn't notice it. The xylem cells themselves are dead at functional maturity. This is important and strange. The cells that conduct water in a mature tree, trachoids and vessel elements in most angiosperms, have lost their cell contents, their nuclei, their organelles. What remains is the cell wall, lignified, rigid, hollow, a tube. The cell died so that it could become a better pipe. The tree's water transport system is built from the carefully arranged corpses of its own cells. I keep expecting this fact to get more attention than it does. It's remarkable. A living system that functions by manufacturing and deploying dead components. But sure, photosynthesis gets all the press. Dissolved minerals travel upward in the xylem stream, calcium, magnesium, potassium, iron, drawn from the soil through root hairs and loaded into the xylem at the root. They arrive at the leaf in the transpiration stream and are deposited there as the water evaporates. Calcium in particular moves almost exclusively in the xylem and is deposited permanently in leaf tissue. It doesn't remobilize easily. When the leaf falls, its calcium leaves with it. This is why calcium deficiency in plants tends to show up in the newest, fastest growing tissue first. The young leaves and shoot tips have high demand but receive xylem water last. They're at the end of the line. If calcium is scarce in the soil, the newest growth gets the least. The xylem system distributes resources, but not equally. Proximity to the engine, the leaf, the transpiring surface, determines priority. At night, when transpiration slows and stomata partially close, the tension in the xylem column decreases. Some cavitated vessels refill as root pressure, a positive pressure that some plants can generate when transpiration is low pushes water back into the system. The tree uses the night to perform a kind of maintenance on its own vascular system. In spring, before the leaves emerge, some deciduous trees generate significant root pressure, enough to push xylem sap upward and out through wounds in the bark. This is why maple syrup is collected in late winter and early spring before leaf out. When root pressure is at its seasonal maximum and the xylem sap is still rich with the sugars mobilized from winter starch reserves, the maple is pressurizing its system for the season ahead. The fact that humans figured out how to intercept this process and reduce it to syrup is honestly one of the more creative things our species has done. Right now, in whatever forest exists nearest to where you're listening, the xylem is running at reduced capacity. It's dark, transpiration is low, the tension in the water column has eased slightly. The roots may be refilling vessels that cavitated during the heat of the afternoon. The phloem is still moving and sugars flowing downward toward root tips that don't stop needing carbon just because the light went out. Two systems, opposite directions, the same tree, one dead, one alive, both necessary. And the carbon moving downward through the phloem right now, made from atmospheric carbon dioxide this afternoon, fixed by chloroplasts in leaves that are currently closed and dark, is on its way to the roots, where it will feed fungi, which will extend through the soil toward minerals, which will return to the leaves, eventually. The whole system is a long way of saying what goes up must come down, and what comes down was always trying to go somewhere else. There is a zone around every root that most people will never think about. Not because it's obscure, because it's invisible, because it exists at a scale and in a medium, dark, wet, compressed, that doesn't offer itself to casual observation. You'd need a microscope and considerable patience and probably a tolerance for the smell of soil, which is its own acquired taste. The zone is called the rhizosphere, and it is, per cubic centimetre, among the most biologically active environments on the planet. Roots don't just sit in soil, they change it. Every growing root tip is secreting, constantly, as a function of normal metabolism, a range of compounds into the immediate surrounding soil, sugars, amino acids, organic acids, enzymes, mucilage, compounds that alter the pH of the soil within a few millimeters of the root surface, compounds that dissolve mineral forms of phosphorus and iron that would otherwise be chemically locked and biologically unavailable. The tree is not trying to do any of this in any intentional sense. It's managing its own internal chemistry, adjusting the electrochemical environment at the root tip to facilitate mineral uptake, lubricating the path of the growing root through the soil with mucilage, releasing metabolic byproducts that simply exit through the root surface because that's where they end up. The soil around the root gets transformed as a side effect. Here's where I want to slow down, because I think this is genuinely underappreciated. The rhizosphere isn't just chemically different from bulk soil, the soil a few centimeters away from any root. It's biologically different by an order of magnitude. Bacterial densities in the rhizosphere can be 10 to 100 times higher than in the surrounding soil, not 10% higher, 10 to 100 times. The carbon compounds leaking from the root surface represent a food source concentrated enough to support microbial populations that bulk soil simply cannot sustain. These bacteria are not passive recipients. They are metabolically active, producing their own compounds, some of which the root can absorb. Fix nitrogen from nitrogen-fixing bacteria, plant growth hormones produced by certain rhizosphere species, antibiotics that suppress root pathogens. The root changed the chemistry attracted the bacteria. The bacteria changed the chemistry further in ways that benefit the root. Nobody planned this. It assembled itself around the coincidence of need and proximity. I've been sitting here for about 20 minutes trying to figure out how to explain what I find so striking about the rhizosphere, and I keep getting distracted by the fact that my nose has chosen tonight of all nights to run a full systems audit. But here's the thing. The rhizosphere is essentially what happens when you leak. Not as metaphor, literally. The root leaks sugars, acids, enzymes, and that leaking creates an ecosystem. The leaking is not a flaw. It is the mechanism. The tree's inability to perfectly contain its own chemistry is precisely what makes the zone around its roots more alive than anywhere else in the soil. I find that quietly important. The productivity isn't despite the leakage, it's because of it. Root exudate composition isn't constant. It changes depending on what the tree needs. When phosphorus is scarce, roots increase their secretion of organic acids, citric acid, malic acid, oxalic acid, that solubilize mineral-bound phosphorus by chelating the calcium or iron it's attached to, releasing it into soil solution where the root can absorb it. When the tree is under pathogen attack, some root exudates shift toward compounds that recruit specific beneficial bacterial species known to suppress pathogens. The tree is, in a functional sense, farming its own soil, selectively enriching the microbial community in its rhizosphere by releasing compounds that favor specific organisms over others. It's not conscious farming, but the outcome is structured, directional, and responsive to what the tree currently needs. This is where the niche analogy comes in, and I'm aware it's slightly unhinged, but stay with me. There's a concept in fermentation, specifically in natural wine and sourdough, called terroir of process. The idea that the microbial community present during fermentation is shaped by the environment, by the substrate, by the accumulated history of what has been fermented in that place before. You don't just add microbes. You cultivate conditions. The conditions select the microbes. The microbes produce the outcome. The rhizosphere is terroir. The root is the winemaker that doesn't know it's making wine. The soil that exists immediately around a root is not generic soil. It is soil that has been cultivated unintentionally, continuously, by the specific chemistry of that specific root system in that specific location for however long that root has been growing there. Two trees of the same species, 10 meters apart, will have meaningfully different rhizosphere communities because their root chemistries have been shaping their local soil in slightly different ways, in slightly different soil conditions for years. Every root creates its own particular world. The carbon that exits the root as exudate is carbon the tree will never see again. Unlike the sugars that travel downward through the phloem to mycorrhizal partners, where there is at least a mineral return, rhizosphere exudates are largely a one-way transfer. The tree releases carbon into the soil and receives not carbon back, but services, a better mineral environment, pathogen suppression, a microbial community that makes the soil around the root more productive than it would otherwise be, carbon for services, an economy running entirely on biological coincidence. The microbes that consume root exudates don't stay put. They are eaten. Protozoa, single-celled organisms that graze on bacteria, consume enormous quantities of rhizosphere bacteria and release the nitrogen locked in bacterial cell proteins as ammonium in a form the root can directly absorb. This is called the microbial loop, and it is one of the more elegant nutrient cycling mechanisms in soil biology. The root feeds the bacteria get eaten, the eating releases nitrogen, the nitrogen feeds the root. The root is farming, but the harvest comes back as something different from what was planted. There's a specific kind of tired I've hit tonight, not sleepy exactly, just the kind where everything feels slightly slower than usual, and you notice things you'd normally move past. I've been thinking about the rhizosphere for the better part of an hour, and I keep landing on the same thing. The most productive zone in the soil is the one immediately around the thing that's alive. Not because of anything the soil did, because of what the living thing couldn't stop releasing. Sloughed root cap cells, the cells that wear away from the root tip as it pushes through soil, are themselves a carbon input to the rhizosphere. Each root tip produces these cells continuously, replacing them as they're lost. The discarded cells don't disappear. They decompose in the immediate vicinity of the root, releasing their carbon and nitrogen into the zone the root is about to grow into. The root is, in a small way, composting ahead of itself, preparing the ground it hasn't reached yet, with material from the ground it just left. Somewhere right now in the dark and the moisture and the compressed silence of forest soil, a root tip is moving forward at a rate too slow to observe directly. Ahead of it, the soil is already being altered by compounds that arrived before the root did. Enzymes secreted by the growing tip, mucilage from the root cap, exudates from the older root tissue behind. The root is never arriving cold. It has been sending its chemistry ahead of itself. And in the soil it's approaching, bacteria that have been living on that chemistry for weeks are already there, already waiting, already making the ground slightly more hospitable than it was before the root decided to come this way. The ground absorbs what the root releases and becomes, because of it, the kind of place a root would want to go. There is a depth in soil where things stop moving. Not immediately, not sharply. It's a gradual withdrawal, the biological activity that dominates the upper horizons thinning out as you go deeper. The microbial community is becoming sparser, the root tips retreating, the fungal threads ending. At some depth specific to each soil and each forest, the frantic cycling of the surface becomes something else entirely, something closer to stillness. And in that stillness, carbon accumulates in a form that most of the living world above it cannot touch. The process that puts carbon here is slow even by soil standards. Dissolved organic matter, compounds released during decomposition in the upper horizons, leached downward with rainfall and snowmelt, moves through the soil profile over years and decades. Most of it is consumed by microbes long before it reaches the mineral horizons. But some fraction, the fraction that resists rapid microbial processing, keeps moving. When it reaches zones where certain minerals are concentrated, iron oxides, aluminum oxides, specific clay minerals, something happens that is chemically mundane and ecologically enormous. The dissolved organic carbon binds, the bonding mechanisms vary. Some organic molecules adsorb onto mineral surfaces through electrostatic attraction. The mineral surface carries a charge, the organic molecule carries an opposite charge, and they come together the way two people at a party gravitationally find the one other person who reads the same obscure thing they do. Other molecules form ligand exchange bonds with iron or aluminum, stronger, more specific, harder to reverse. The organic carbon essentially trades a hydroxyl group for a spot on the mineral surface, and once it's there, it tends to stay. This is mineral-associated organic matter. MAOM in the shorthand that soil scientists use when they have typed it enough times to want an abbreviation. Growing up in Cincinnati, you get a particular relationship with soil that I don't think gets talked about enough. The Ohio River Valley sits on some of the deepest, oldest glacially deposited soils in the Midwest. When I was a kid, digging in the backyard past a certain depth meant hitting this dense, pale clay layer that felt completely different from the dark topsoil above it. Colder, heavier, almost ceramic. I didn't know what I was looking at then. I just knew it felt like a different world. That pale clay layer, that's where mineral-associated organic carbon accumulates. I was literally digging into a carbon archive and didn't know it. The carbon held in mineral associations is old. Radiocarbon dating of MAOM fractions from deep soil horizons routinely returns ages in the hundreds to thousands of years. Not because those carbon atoms have been sitting inert since then, some turnover does occur, but because the average residence time of carbon in these mineral associations is genuinely long, measured in centuries rather than seasons. For context, the carbon cycling through leaves, through the fast decomposition of fresh litter, through the bodies of soil bacteria, that carbon turns over in years, sometimes months. The carbon bound to mineral surfaces in the deep horizon of an old forest soil is operating on a completely different clock. The same forest, different time. What keeps this carbon stable is, primarily, inaccessibility. Microbes that decompose organic matter need physical access to it. Their enzymes need to contact the substrate. Organic carbon bound to mineral surfaces is, in many cases, physically shielded. The mineral surface occupies the space where the enzyme would need to attach. The carbon is there, chemically, but the microbial machinery can't get purchase on it. It's not that the carbon is chemically inert. Under the right conditions, disturbance that exposes the mineral surface, pH changes that break the bonds, shifts in redox chemistry in waterlogged soils, mineral-associated carbon can be released. The stability is conditional. It depends on the soil maintaining the specific conditions under which the carbon was stabilized. Change the conditions, and the archive opens. This is the part where I have to be honest about something that sits uncomfortably with me. The stability of mineral-associated soil carbon, this deep, old, slow carbon that we sometimes discuss as though it's safely locked away, is not unconditional. It never was. We just assumed for a long time that it was, because we were measuring on timescales too short to see it move. More recent work suggests that warming temperatures, changes in soil moisture, and shifts in microbial community composition can destabilize MAOM fractions that were previously considered stable. The deep carbon archive is not a vault. It is more like, and I say this carefully, a filing system that functions perfectly until the filing conditions change. And the filing conditions are changing. I don't say that to introduce anxiety into what is supposed to be a sleep narration. I say it because the biology is what it is, and I think you can hold that truth and still rest. Knowing a thing doesn't require doing something about it right now in the dark at whatever hour this is for you. There are forests where the mineral-associated carbon in the deep soil represents more stored carbon than exists in all the living biomass above it. All the trunks, all the branches, all the roots, all the leaf litter combined. The visible forest is, in terms of carbon, the smaller part of the story. Most of the carbon the forest has ever processed is below it, in the soil, in forms that don't look like anything in particular. No structure, no cell walls, no rings you can count. Just carbon held against mineral surfaces in the dark, in the cold, at a depth where almost nothing alive bothers to go. There's a particular quality to deep soil that I've always found hard to describe accurately. It doesn't smell like surface soil, that rich, almost sweet microbial smell that you get when you turn over a good garden bed. What scientists have identified as geosmin produced by actinobacteria. Deep mineral soil smells different. Cooler, more mineral, less alive in the sensory sense, even though it contains carbon that was once very much alive. It smells like time, which is an absurd thing to say, but I'm saying it anyway, because I've run out of accurate alternatives. The carbon that arrives in the mineral horizon arrived through processes that are invisible from the surface. A raindrop hitting a decomposing leaf, dissolving a small fraction of organic matter, moving downward through the soil profile over weeks, passing through zones of microbial activity that consumed most of it. The surviving fraction reaching a clay surface 10, 20, 30 centimeters down, bonding, staying. That journey from leaf to mineral surface might take years. The carbon that completes it and bonds successfully might remain bonded for centuries. All of that from a single raindrop. The forest above doesn't know this is happening. The trees are managing their canopy carbon, their phloem transport, their mycorrhizal partnerships, their seasonal resorption. The processes that move carbon into mineral associations are below the root zone, below the biological horizon the tree actively manages, operating through chemistry and physics rather than anything the living forest directly controls. The deep soil builds its archive without instruction, without intention, through the accumulated consequence of everything the forest has ever done above it, filtered downward, arriving here in the form of dissolved compounds that happen to bond with surfaces that happen to hold them. In Cincinnati, that pale clay layer I dug into as a kid is still there. Under whatever has been built over it, under whatever soil disturbance the decades have brought, the mineral horizons of the Ohio River Valley still hold their glacially deposited clays, still accumulate whatever dissolved organic carbon manages to reach them, still running the same slow chemistry they were running before the city was a city, before the forest that preceded it was cleared, before anyone thought to ask what was happening down there. The deep soil persists. It holds what it can. It releases what the conditions require it to release. And in the forests that remain, along the Little Miami River, in the remnant old-growth pockets of southwestern Ohio, in the deeper ravines where the trees have been undisturbed long enough to build real soil, the carbon archive is still accumulating, still going deeper, still settling molecule by molecule against surfaces that have been waiting in the dark since before the glacier left. The last light leaves a forest slowly, not all at once. The canopy holds onto it longest, the uppermost leaves still catching the last flat angle of evening sun, while the forest floor has already been dark for an hour. If you have ever stood inside a forest at dusk and looked up, you know this. The sky above the canopy still pale, almost luminous. Everything below it, the trunks, the understory, the ground, already in a different kind of time. That gradient, that's where the forest carbon day ends. By the time full dark arrives, the accounting has already shifted. The chloroplasts in every leaf have stopped fixing carbon. Not winding down, stopped. Photosynthesis is a light-dependent process in the most literal sense, and when the photons stop arriving, the reactions that fix atmospheric carbon dioxide into sugar simply cease. The enzymes are still there. The chloroplasts are still there. Everything is in place for tomorrow. But tonight, nothing is being made. What remains is everything that was made during the day. Sugars sitting in leaf cells, waiting to be loaded into phloem. Starch granules that formed in chloroplasts during peak photosynthesis. A temporary storage form the leaf uses when it's producing sugar faster than the phloem can export it. Carbon in the structural compounds of cells that divided today. Walls that thickened. Proteins that were assembled. The day's fixation is distributed through the tree in various forms, at various stages of being moved or used or stored. None of it has disappeared. It has just stopped being made. I want to tell you something about recording this chapter specifically. It's late, later than I intended. My cold has crossed into that particular territory where you feel almost human again, but your body is still clearly running some background repair process that it hasn't told you about. Cincinnati in February has a specific quality of darkness, the kind that comes with low cloud cover and no moon, and a quiet that feels thick rather than empty. I've had the window cracked the entire time I've been recording, and there is something, I can't name it exactly, about talking about what a forest holds at night while it is, in fact, night. And there are trees outside this window doing exactly what I'm describing. The carbon they fixed today is in there somewhere, moving, settling, being used. I find that genuinely comforting in a way I don't entirely know how to explain. The phloem is still running. This is worth saying again because it keeps mattering. The phloem doesn't stop at dark. Sugars produced today are moving downward right now toward roots that need them, toward mycorrhizal connections that are running their own quiet chemistry in soil that is degrees cooler than the air above it. The day's photosynthesis is still doing work. It will still be doing work at 3 in the morning. The carbon doesn't stop moving just because the making of it stopped. In the soil, the microbial communities are running their overnight metabolisms. Bacteria that spent the day reproducing on the flush of root exudates released during peak root activity are now in slower phases. Fungal hyphae are extending into dew-moistened surface litter. The soil arthropods, springtails, mites, small beetles that spend daylight hours deeper in the soil profile are moving toward the surface, feeding on fungal threads and decomposing material in the layer of organic matter just above the mineral soil. The forest floor at night is not quiet exactly. It's just quiet at the wrong scale to hear. Here is an opinion I hold with moderate confidence and will state as such. The carbon balance of a forest on any given day, whether it fixed more than it released or released more than it fixed, is one of the more quietly consequential numbers in ecology, and almost nobody outside of a narrow research community thinks about it regularly. Not because people don't care, because it's invisible, because the accounting happens in gases and dissolved compounds and microbial metabolisms that leave no trace you can point to at the end of the day. The forest did something today, something measurable. The number exists. Nobody outside of a flux tower dataset will ever know what it was. There's a technology, eddy covariance flux towers, that measures exactly this. Instruments placed above the canopy that continuously measure the concentration and movement of carbon dioxide in the air, calculating the net exchange between the forest and the atmosphere in near real time. Some of these towers have been running for decades. They exist in forests on every continent, quietly measuring the daily carbon arithmetic of the ecosystems beneath them. The data they produce is some of the most important long-term ecological data we have. Most people have never heard of them. They look like weather monitoring equipment. They stand above the canopy, usually on a metal mast, instrumented and unobtrusive, watching what the forest does. I think about them sometime, these towers standing in the dark above sleeping forests, measuring, patient in a way that requires no patience because they're instruments, not minds, but patient in the functional sense, still there, still measuring, still accumulating the record. There is a version of tonight happening in forests across every time zone right now. In the Appalachians, a few hours east of Cincinnati, the same post-dusk carbon shift happened hours ago. In the Amazon, where it's still afternoon, the canopy is still running full photosynthesis, still building the sugars that will be moving downward through phloem by the time darkness arrives there. In the old-growth forests of the Pacific Northwest, The Douglas firs that have been fixing carbon for centuries are doing the same overnight accounting they've done every night since before anyone alive was born. The same chemistry, different forests, different clocks, all of it running simultaneously, all of it connected through an atmosphere that doesn't recognize any of the borders we've drawn across it. What the forest holds tonight is today's work. Some of it will be used by mourning, consumed by root respiration, incorporated into new cell walls, transferred to mycorrhizal partners, released back into the atmosphere through the simple act of staying alive. Some of it will stay, added to the pool of stored carbon in wood, in stable soil compounds, in the slow accumulation of material that the forest has been building since the first tree took root in this particular soil. Each day adds something. Each night determines what gets kept. The light goes. The keeping begins. This is where the forest's carbon day ends, not with a conclusion, but with the redistribution. Everything that was made is now somewhere in the system, moving toward wherever it's needed, or settling into a form that will hold it until it's needed, or being released back into the air as the simple metabolic cost of all this living. Nothing was wasted. That's not sentimentality, it's chemistry. Every carbon atom that entered this forest today will be accounted for in some form, in some location, at some time scale. The forest doesn't lose things, it just changes where they are. And that, I think, is a reasonable place to rest. Not because the story is finished, it isn't. The carbon is still moving, the phloem is still running, the soil is still doing its slow arithmetic beneath the roots. The story doesn't finish. It just continues at a pace and in a medium that doesn't require your attention tonight. You've been here through 17 chapters, through canopy and soil and fungal networks and fire and the slow patience of mineral horizons, and I'm genuinely grateful for that. This channel, The Sleepy Biologist, exists because people like you keep coming back to it. Keep sharing it with someone who needs a quiet place to land at the end of the day. If tonight's video helped you rest a little better, if something in the biology landed in a way that felt worth keeping, the like button is there, and it matters more than it probably should for something so small. If you're new here, hello, there are many more nights like this one waiting for you. Subscribing means you'll know when the next one arrives. To the channel members and everyone who has sent a super thanks, you are why this space stays alive. Genuinely, not as a phrase, as a fact, and if you want to leave a comment about what stayed with you tonight, what part of the forest you're still thinking about, I read them, all of them. Slowly, the way forests do most things. My cold is still here, the window is still cracked. Somewhere outside, the trees are doing their overnight accounting, completely indifferent to whether anyone is watching. I find that comforting too. Good night.